Welcome to Nefertari Audio Genetics. Today's topic is genome editing versus genetic engineering. These two approaches are often discussed together because both are used to modify genetic information. However, they are not the same. Understanding the difference is important not only for understanding modern biotechnology, but also for choosing the right approach for the right biological question. Let's begin with the basics. What are they? Genetic engineering is a broad term that describes the deliberate modification of an organism's genome, often by introducing new genetic material. While genome editing is a more targeted approach, instead of adding a new gene, it modifies DNA at a specific location within the genome, allowing precise changes at existing genetic information. How do they differ? The first major difference is where the change occurs. In many classical genetic engineering approaches, introduce DNA integrates at random or semi-random locations within the genome. While in genome editing, by contrast, it is designed to make changes at predefined genomic sites. The second difference is the type of genetic change. In genetic engineering, it's commonly used to introduce a new function or trait, whereas genome editing is generally used to modify, remove, or fine-tuning genes that are already present. These differences also influence the workflow. Because genome editing targets a specific location, it is often streamlines the development process when precise modifications are required. Genetic engineering, however, remains the preferred option when the objective is to introduce an entirely new biological function. Now let's discuss the common concerns about genetic engineering. There are three main concerns that are frequently discussed when talking about classical genetic engineering. The first is random insertion. Because the introduced DNA may integrate into different genomic locations, the gene expression can vary, and in rare cases, an existing gene or regulatory region may be disrupted. Scientists address this by screening multiple independent events, identifying insertion sites, and selecting stable, well-characterized lines. The second concern is foreign DNA. When genes originate from another species, questions may arise about safety or ethics. Scientists evaluate every product individually by assessing the source of the gene, the properties of the encoded protein, and any potential of allergenicity or toxicity. The third concern involves selectable marker genes. Modern molecular techniques allow researchers to remove these markers after selection, eliminate them from breeding, or use marker-free systems, ensuring that the final product contains only the intended genetic trait. So, which approach should you choose? The answer depends entirely on the biological question you are trying to answer. If your goal is to introduce an entirely new trait or biological pathway, genetic engineering may be the appropriate choice. If your objective is to investigate gene function, reproduce a naturally occurring variant, or make precise changes to an existing gene, genome editing is often the better approach. Rather than competing technologies, genome editing and genetic engineering are complementary tools. Each has its own strengths, limitations, and applications. The important question is not which technology is better, but which one is best suited to answer your scientific question. Thank you for listening to Nefertari Audio Genetics. Until next time,